Hello friends, today we are going to study about composition, structure and function of carbohydrates part 4. Today we are going to study about polysaccharides. What are polysaccharides? Polysaccharides are the carbohydrates that consist of more than 10 molecules of sugar. In nature, carbohydrates are mainly found in the form of polysaccharide only. Now these are also known as glycans. Polysaccharides are also known as glycans. Okay. Polysaccharides can be of two types, homopolysaccharide and heteropolysaccharide. Homo means same or single variety. When a polysaccharide consists of single type of monomer, then we call it as a homopolysaccharide. Hetero means different. When polysaccharide is composed of different types of monomers, then we call it as a heteropolysaccharide. Homopolysaccharide function as storage form of energy and also as structural element. These are the two types of function of a homopolysaccharide. They function as storage energy and structural element. For storage, they have two types of polysaccharides. One is glycogen found in animals and starch found in plants. For structural, chitin is the homopolysaccharide found in animals and cellulose present in plants. Okay, so homopolysaccharides means consist of single monomer of single type of monomers. They function as storage and structural element. For storage, we have glycogen homopolysaccharide in animals and we have starch for plants. In case of structural element, we have chitin in animals and cellulose in plants. Then next is heteropolysaccharide. Heteropolysaccharide means it consists of different types of monomers and they mainly function as a extracellular support. Heteropolysaccharide, whether it is in plants, whether it is in animals or in bacteria, in any organism, they function as an extracellular support. Now let's study about the first carbohydrate, starch. It is a storage carbohydrate. Starch is a storage form of energy, stored energy in plants. Now, this is the structure of starch. Before that, starch occur intracellularly as a large granules and these are heavily hydrated. Why starch are heavily hydrated? Because they have many exposed OH group. These OH group can form hydrogen bond with surrounding water molecules. Therefore, starch are heavily hydrated. Now starch composed of amylose and amylopectin. Now see here, this is the structure of amylose and this is the structure of amylopectin. What you can see in the structure, starch is a homopolysaccharide, right? So here also glucose, here also glucose. Starch is a polymer of glucose monomers. This bond, definitely a glycosidic bond. This is C1, this is C4. Therefore amylose have 1 to 4 glycosidic bond. As because this is alpha D glucose, so we can say this is amylose contain alpha 1,4 glycosidic bond. In case of amylopectin, this is carbon number 1. This is carbon number 6. So amylopectin contains alpha 1 to 6 glycosidic bond. Now it also contains alpha 1 to 4 glycosidic bond. Why? Because you can see dot dot here. That means chain length increase this side as well. So that means it consists of many glucose unit, not one. So all the glucose unit which are linear will have alpha 1,4 linkage, glycosidic linkage. Wherever branching will be there, that point will be alpha 1,6 linkage. So starch consists of amylose which have alpha 1,4 glycosidic bond and it also consists of amylopectin which have alpha 1,4 and alpha 1,6 glycosidic bond. All the glucose will be connected to each other by alpha 1,4 glycosidic bond. Wherever branching will be there, at that point, one glucose will be connected with the other glucose through alpha 1,6 glycosidic bond. Now, branching, the position is after 24 to 30 residue approximately, there will be one branch. So, starch is a polymer of glucose unit, but glucose are arranged in the form of amylose and amylopectin. In case of amylose, it is a linear chain of glucose. Therefore, the bond is alpha 1,4 glycosidic bond. In case of amylopectin, it is a branched chain of glucose. So, wherever branching is there, the bond is alpha 1,6. Apart from that, wherever the linear chain is present, the bond is alpha 1,4. Next carbohydrate is, or next polysaccharide is glycogen. It is also a storage polysaccharide, but in case of animals. Glycogen is almost like amylopectin only. So wherever this dot dot, that means linear chain. Wherever linear chain will be there, the bond will be alpha 1,4. Wherever branching will be there, the bond will be alpha 1,6. In case of glycogen, the branching is much more as compared to the starch. In case of starch, branching occur after every 24 to 30 residue. But in case of glycogen, branching occurs after 8 to 12 residue. Glycogen also occur intracellularly as large granules. It is also heavily hydrated. Reason is the same, because it has many exposed OH groups. These OH groups can form bond with surrounding water molecules. Glycogen is also a polymer of D-glucose units. It is like amylopectin. Wherever branching is there, alpha 1,6 bond. Wherever linear chain is there, alpha 1,4 bond. And branching occurs after every 8 to 12 residue. Therefore, glycogen is more compact compared to starch. Now, if you see this branching, suppose this is one glucose unit, second glucose unit, third glucose unit. What will be this bond? Alpha 1,4, alpha 1,4. Branching came. Here again, alpha 1,6. Again, suppose it is a linear. This branching came. Here, alpha 1,6. Now, we already studied sugar molecule can have a reducing or a non-reducing end. What kind of sugar molecule? Disaccharides and polysaccharides can have a reducing as well as non-reducing end. Now, in case of this glucose and this glucose, here C1 is involved, here C4 is involved. To be a reducing end, because it is an aldose, which carbon should be free? The first carbon. Why? Because this first carbon has CHO group, and this only can take oxygen and convert into COOH. We already studied this in the last videos. Those who have not seen those videos, I have provided the link at the end of this video. You can go and check them once. So here C1 is involved in glycosidic bond formation with C4. This glucose is again connected with the next glucose. So now the C1 carbon, the first carbon of this glucose is attached with the fourth carbon of this glucose. Now for this glucose, again the C1 is free, but now this C1 is branched with this particular glucose with alpha 1,6. The sixth carbon of this sugar is bonded with first carbon of the above sugar. So here also C1 is attached in glycosidic bond. So from here to here, doesn't matter how long the chain is, all the C1 positions are occupied in bond formation. So no reducing ends are present here. Now come here. Here also same. For this glucose, the sixth carbon is occupied and the first carbon is occupied with the next glucose, because here again alpha 1,4 bond. So all the ends are non-reducing because first carbon is not available. First carbon is not free. But suppose here this is the compound's last glucose. For this, the C1 is free because this is the last glucose molecule for glycogen. So no more glucose is attached with this. So for this glucose, the C1 is free. So that means in case of glycogen, it contains many non-reducing end and only one reducing end. So glycogen contains many non-reducing end and only one reducing end. Let's revise it one more time. For this carbon, for this sugar, first sugar, it is bonded with the second sugar by alpha 1,4 glycosidic linkage. So for the first sugar, first carbon is occupied. Now for the second sugar, fourth carbon is occupied with the first sugar and first carbon is occupied with the third sugar, because here also 1,4. So here also first carbon is occupied. Now for this third sugar, fourth carbon is occupied with the second sugar and first carbon is occupied with the branched sugar at sixth position. So here also first carbon is occupied. Now for this sugar, sixth position is occupied with the first carbon of the above sugar. But in case of linear, again the first carbon here is bonded with fourth carbon of the next sugar. So here also first carbon is not occupied. Again for this sugar, first carbon, fourth with this carbon. So this process continues. So for none of the glucose, the first carbon is free. Because first carbon contains functional group aldehyde, and this carbon only takes oxygen and gets converted into COOH, and hence sugar is known as reducing sugar. So here only the last glucose, to which no other glucose is attached, for that glucose only the C1 is free. Rest, for all the glucose, the C1 is not free. Hence, glycogen contains many non-reducing end, but only one reducing end. Why in a cell, glucose cannot be stored in the form of glucose only? What is the need to convert it into starch or glycogen? Now, in a cell, the concentration of glycogen is 0.01 μm. This concentration is equivalent with the glucose concentration 0.4 molar. If instead of glycogen, glucose is present in the cell, it increases the osmolarity of the cell. Whereas glycogen does not increase osmolarity. It contributes very little for the osmolarity. So if in place of glycogen, glucose is present, it will increase the osmolarity of the cell. And what will happen? The water will start flowing in and cell will burst. Hence, glucose always converts into a polymeric form to be stored inside the cell. Starch in case of plants and glycogen in case of animals. So, because they contribute very little for the osmolarity, so it is stored in the form of starch or glycogen. If it is present in the form of glucose, it will increase the osmolarity of the cell, and because of which the water will start flowing in and the cell will swell and burst out. Next is dextran. These are bacterial and yeast polysaccharide. They are also branched like a myelopectin, but the bond involved can be alpha 1,6, alpha 1,3, and sometimes alpha 1,2 as well as alpha 1,4. So dextran are bacterial and yeast polysaccharide. They are also branched. The branching point is alpha 1,6, can be alpha 1,3, can be alpha 1,2, and also alpha 1,4. These are also found in the biofilm, which is the cause of dental plaque. Next is cellulose. So if you see the structure of cellulose, it is also a polymer of glucose monomers. Cellulose are fibrous, they are tough, and they are water insoluble. Why they are water insoluble? Because according to X-ray study, it was found that cellulose are flat structure in which successive glucose are turned over 180 degree, due to which a hydrogen bond formed between C3OH of one glucose with the O of the fifth carbon. So what happens? The cellulose are flat structured and they form a sheet in which the successive glucose are turned 180 degree in such a way that one glucose is hydrogen bonded with the other glucose. The hydrogen bond is formed between C3OH group of one glucose with the O of the fifth carbon of the second glucose or of the next glucose. Because of this extensive hydrogen bonding, cellulose become insoluble for water. Next is, these are also linear, unbranched. The glucose unit present are 10,000 to 15,000. The difference is with, if you see the structure, it is looking like amylose only because it is a linear chain. Amylose has linear chain of glucose monomers. So this also, cellulose also contains linear chain of glucose residues. So it is like amylose. The only difference is in case of amylose, it is alpha 1,4 bond. That means the glucose are present in alpha orientation, alpha anomeric form. But here the bond is beta 1,4 glycosidic bond. The glucose are present in beta anomeric form. These are found in cell wall of plants. Animal cannot digest cellulose because it is in beta form. So the bond is beta glycosidic bond. Animals or we don't have enzyme because of which we cannot degrade cellulose. These are the most abundant polysaccharides in nature. Next is chitin. Now if you see the structure of chitin, it is almost like cellulose. The difference is it contains N-acetyl glucosamine. N-acetyl amine group is present along with glucose. So it is a polymer of N-acetyl glucosamine monomers. Rest all same like cellulose. These are also linear, unbranched. beta 1,4 glycosidic linkage. These are found in the exoskeleton of insects, cell wall of fungi and algae. Like cellulose, we cannot digest chitin as well because it is also in the beta form. These are the second most abundant polysaccharide in nature. The first most abundant polysaccharide is cellulose and the second most abundant polysaccharide is chitin. So let's revise what all we studied. We studied about homopolysaccharide and heteropolysaccharide. Homopolysaccharide function as storage form of energy and structural element. Hetero mainly extracellular support. Today we studied about homopolysaccharides. For storage we have starch and glycogen. Starch in plants, glycogen in animals. For structural we have cellulose and chitin. Chitin in animals, cellulose in plants. Starch has amylose and amylopectin. Amylose has alpha 1,4 linkage, amylopectin alpha 1,6. Glycogen is like amylopectin of starch. Cellulose is like amylose of starch. The difference is it is in beta conformation. Amylose is in alpha conformation and that's why we cannot degrade cellulose because we don't have enzyme to degrade beta glycosidic bond. Chitin, it is also like amylose. The difference is it contains N-acetyl glucosamine monomers. The most abundant polysaccharide is cellulose. Next abundant is chitin. Chitin is found as an exoskeleton in insect and cell wall of fungi. Cellulose is found in the plant as a cell wall component, where they found along with lignin and other polysaccharide. And therefore, it is very difficult to convert cellulose into biofuel because they are very strongly attached with other polysaccharides and lignin in the plant cell wall. Therefore, it is very difficult to convert cellulose into biofuel, though it is present in very abundant. Glycogen is a stored carbohydrate of animals. Starch is a stored carbohydrate of plants. Now, one question from my end for you all. How many reducing ends are in a molecule of glycogen that contain 10,000 residues with a branch at every 10 residue? You have to tell me how many reducing ends a glycogen molecule can contain, that is 10,000 residues long and branch at every 10 residue. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.